Well, thank you, Carol, for that very kind introduction, and welcome to all of you. Thank you very much for coming along to this morning's morning tea. I hope we can tell you about some interesting science, Michelle and I. Michelle, the much better looking one, is coming after me, of course. As Carol mentioned, my expertise is in the immunology area, and particularly how we make antibodies and how that's controlled. But one of the ways we do that in particular is by using animal models, mouse models, which are a fantastic model for lots of things in studying how the body works. Of course, we are able to study in detail in mice lots of things we can't do with people, but the important thing is we have a back and forward between these two systems. And something I've been involved with for a long time is making genetically modified versions of mice, which are a really powerful way of picking apart how the body works and what goes wrong in disease. And in particular, what I want to tell you about today is a very new, for those who actually managed to catch that Ask the Doctor episode, a large feature of that was this new technology called CRISPR. And I want to tell you a bit about that and how it gels in with our efforts in genomics, in genome sequencing that Carol mentioned, tying in Alan's story, which is just a great one, and how we're extending stories like that to make further breakthroughs with a whole range of diseases, basically apply not just to the immune system, but to everything. Gene editing, that's what CRISPR does. It's the ability to change genes, and whilst we've always been able to do that to a degree, the revolution here is that it's so much quicker, so much, well, fast is the same thing, so much cheaper and manipulable than anything we've had before. So I hope that's the message I can give you with, and all the promising things we'll be able to do with it. So genetic diversity. So I've put up everything on the life on Earth is all driven by your genes. Obviously, there's other inputs. There's the environment, but your genes are what make up the different species and account for lots of all our differences between bacteria and viruses and us and everything. Our genes make us what we are, and so even within a particular species, like your kiwi fruit up the top left there. There's not just a kiwi fruit, but there's all different types of kiwi fruit, and the reason they're different is because of their genes. Sweet potatoes as well. I mean, I couldn't believe there were that many types of sweet potatoes, but all that's because of genetic diversity and people selecting that actually over time by saying, well, I like the look of that kiwi fruit or that sweet potato, let's pick that one. And the reason it arose in the first place was because there was a change in its genes, which made it look and perhaps taste different, or maybe be cultivated in a different way. Of course, this applies to mammals as well. Our friends the dogs are another example where we've selected for a lot of, we've had natural changes in genes over time. And of course, people look like this dog, like that dog, and there's been a high pressure to pick all sorts of different dogs. And of course, we are extremely diverse bunch in terms of our genes and how we've evolved over time. Very similar in most ways, but of course we do have differences that explain a lot of the visual and I guess our susceptibility to diseases or health and other things as well. So we're talking about genes. Carol mentioned we're now have a facility to look at the DNA, the genome from single cells. So every cell in the body that's meant to be a cell looks more like a kind of strange mouse or something, or some mutant. But that's a particular type of flagellated cell inside the body, and of course each cell has within it a nucleus, and within the nucleus carries 46 chromosomes, and within those chromosomes are the information that program how the cells work, how they respond to being what they are inside the body. And of course your DNA is made up of genes. Your genes are made up of DNA, this famous double helix, like our staircase next door. These little bars across here are basically little chemical groups that we represent by 4 letters, A, T, G, and C. And so this little stretch of DNA here may be about 20 of those letters, and each of our cells has 3 billion of those. So there's an incredible amount of information packed into every cell, and all of it's coded in these individual letters wrapped up in the double helix, as I'm sure many of you will know. So over time, we didn't even know DNA existed really, or to carry the genetic information. That was Watson and Crick and Rosalind Franklin who found that out in the 50s. And of course, since then, we've not only found out what DNA is, but we now have got to the point, as those of you have heard John Maddock talk so eloquently about how we can actually access all that information incredibly quickly for the price of $1,000 to $2,000 now, and so rapidly compared to $15 billion in 10 years, even just 15 years ago. So whilst our DNA serves us very well, it can pick up changes which may cause us problems in one way or another. And so these are our chromosomes floating around in the DNA in the nucleus. And so this hot spot here is it's actually not what it looks like. It's just highlighting that there's been a change in the chromosome, and what that means if you pick up a mutation in a chromosome, and that can happen in a number of ways. It can just happen by chance. DNA replication is not 100% faithful. It's occasionally going to make a mistake, and so whilst there are things which speed up that process, perhaps like smoke and like ultraviolet light and stuff, it does happen as a matter of course as well. And so it can be as little as a change of one of these letters in DNA. And so in the wrong place, even just changing one of these A, G, C's and T's can make all the difference into whether you have a particular disease or not. So when we see diseases which perhaps we can't explain, but certainly ones which seem to pass through a family, there's a strong chance that disease may be wrapped up into some genetic change which has passed through that family. I mean, you can always be the first one to pick up a mutation as well, but particularly when we see a family where you have an inheritance, there's a strong indication that something's happening in a genetic sense. So how do you sort out what that is and what you can do about it? So one of the high-tech machines Carol was mentioning was these guys, the High-seq whole genome DNA sequencers. So they're also upstairs in level 10, I think. So we've got a whole lot of very powerful machinery up there these days. And so John's objective, dream really, in terms of securing this very high-tech, high-throughput facility was to be able to process many genomes from people who may have thought to have a genetic disease of some sort, and try and sort out what those are. So, if you've got one base change out of 3 billion, it's really worse than a needle in a haystack. If you have no way, where do you start? The first thing you can do is you could pick places where you think it might be happening, but the way to give yourself the most chance of finding it is to take that genome, look at all 3 billion letters, and okay, what looks like it's not right? What looks like it's different to? Nothing's really normal, okay? But there are things that we can pinpoint as being likely to be causing a particular disease, particularly if we know about a bit about that disease and what might be going wrong. So the process then is that, for instance, with the case of Alan, which is just such a great example of the power of this technology, is Alan was over at the kids' hospital over here at Randwick, had this autoimmune condition which no one could explain, and it was really quite debilitating. And so his immune system was destroying all his red blood cells and platelets. He would get these spontaneous bleeds. And it was clear his immune system was acting out, but it wasn't clear why. So he was a great candidate. No, in his case, there was no history in the family, and it turned out that he had picked up from his mum and his dad, just very unfortunately, two bad copies of one gene. So you need actually two copies, usually, of a bad gene because usually if you've got a good copy and a bad copy, the good copy can save you, and that was the case with his parents. But he was unlucky enough to get two of these very rare mutations in this one particular gene. And so that was apparent once his genome got sequenced, and with a bit of work. So Chris Goodnow, who was mentioned before and who was on the show, was really instrumental in looking at this. Being an expert immunologist, he was able to dive into the research, into his connections with people in the States who are working in this area, and it's known that particular gene was important for basically being a brake on the immune system. So we normally got to stop the immune system from getting too excited if it's attacking a virus or something, because it has so much power. and keep it focused on what needs to attack and not to attack us. But so in Alan, this brake was missing and this particular protein wasn't working. So it turned out that there was a therapy in which essentially this protein could be injected back into Alan with the hope that that might put the brake back on his immune system. And in fact, it just worked incredibly well. And so he went from being someone who couldn't essentially move around for the sake of these bleeds to again, if you saw the show, he was jumping around and he was having sword fights with the presenters and stuff. So that's an incredible success story, and just showing the power of the investigation, and without previous research into how the immune system works, you're nowhere really. I mean, you might see there's a DNA change, but unless you know what that gene's doing from other work, you can't really predict what's going to happen. So, and that was a good case. I mean, that was in that case, let me say, there was a mutation, and it was with a bit of work and knowledge, high-level knowledge, it was possible to know what to do for Alan. However, that might not always be the case. I mean, you might have a disease where there's a mutation and you think, okay, well, clearly there's a mutation here, but is this what's causing that? There's a number of other changes in here and maybe they're causing it. How do we sort out what's going on? So this is where CRISPR comes in one respect. I'm not sure how well you can see that, but CRISPR stands for, I won't tell you what it stands for, but yes, I will. Clustered, regularly interspersed non-palindromic sequences, or something like that. So it was basically identified in bacteria. I mean, it was one of the people will be receiving Nobel Prizes any time now for this. It's just amazing. Could bacteria use this system to attack viruses? It's like a bacterial immune system, and so they were able to send this protein called Cas9, and what Cas9 can do, that's Cas9 binding to a bit of DNA, and it's about to chop it in half. So what do you want to do that for? Well, what bacteria can do is actually direct this Cas9 protein, and when a virus infects them, this Cas9 protein can specifically recognize the viral DNA and start chopping it up. And so it's a bit like an antibody in the immune system. It's specifically attacking the invaders as it comes in. So, which is quite amazing, and on its own, that's quite an incredible bit of evolution. But what these incredibly smart people in Berkeley and in Boston sort of simultaneously realised that if you can actually direct this protein to cut DNA, that's really a first step in being able to edit that piece of DNA, because you can repair the DNA very efficiently then, and so introduce a change into that site, or perhaps even correct a change that was already in there, and so. What we do then, and this is we set up a facility here a couple of years ago. It's called MEGA, Mouse Engineering Garvan ABR, and ABR is our animal facility down in Mossvale. And so what we do is in part we act as a next stopping point. Once we have a gene identified in someone with a disease, and it's not certain what that gene's doing, well, what we can do is take that mutation and say, okay, we have it in a very complicated human with all these other changes. What if we just take that one specific change and put it into an inbred mouse where all the mice are essentially the same genomes, okay? There's not the purposefully generated by brother-sister matings over many, many decades. So they've essentially a whole bunch of twins, if you like, clones. And so if we change one gene in one of those mice, then we can compare it to the other mice and say, well, okay, we've got a very controlled system now to really interrogate what that mutation's doing. So essentially what we do is there's a gene in a mouse, and what we do is just micro-inject into a very early stage embryo, just before that's the two nuclei from the sperm and the egg, and we were able to micro-inject in just the DNA, the CRISPR-Cas9 machinery, the DNA and RNA, and what it's able to do is just seek out the particular gene we want in that embryo and introduce a modification. And so once that embryo grows up, it's the same as its parent, but it's got this one mutation in it. And so what we do then is we have a perfectly controlled system, as I say, and so we can take the original mice and the mutant mice and then say, the first question we ask is, okay, we think this mutation is causing a disease. Does this mouse have a disease? Or does it have something that's looking like the disease? If it does, we've verified that particular gene is a large part responsible for the disease in the original patient. And any patients which come along in the future with the same types of mutations. So that's great, but then that may be enough to allow us to feed back treatments to the patient. It may be just a starting point though. If it's really something we don't understand yet, what this gives us is a fantastic model for future research. We can find out what this mutation is specifically doing to, say, if you have an immune system or a liver problem or whatever, what this mutation's doing in a molecular detail, cellular detail, and then based on that, increasing our knowledge about these things and how they work. But then it's a great system for saying, okay, we have a mouse. It's got the same problem as patients with this treatment. There's a bunch of things that we could do. Which one do we take into the patient? Well, this is a great way for testing some therapies very rapidly on a large scale, without having to do massive, expensive clinical trials. I mean, they come later, but this is a fantastic way for running the candidates through much more quickly and much more cheaply. So, this is just a quick example. So that's a DNA sequence as we take it from a standard DNA sequencing read. And all these peaks are just your different letters of the code. And what was found in this case was there was a particular autoimmune disease linked to a gene where there was two mutations. This C was changed to a T, this G was changed to an A. And we were able to use the CRISPR-Cas9 process to generate a mouse which was a faithful reproduction of that. And that this is two copies of the gene, and you can see on one copy the C has been changed to the T and the G has been changed to the A. So we have a perfect copy then, and already that's actually shown us some really unexpected problems in how the immune system is dealing with cells which might attack itself. And so that's sort of an example of the next level down, where we've gone into the mouse and we're now developing some knowledge and potential therapies to treat this particular disease back in the patients. Okay, so then just a summary. Sorry if I've gone a bit over. So CRISPR-Cas9, I'd be happy to, if people want to know more details about how that works. It is quite involved, but it is an incredibly powerful research tool, first and foremost for us, I guess, and it allows us to do this very rapid and real-life assessment of mutations we find in patients. It also has the ability to correct mutations, and so that's been done in mice with genetic diseases, so that's perfectly possible. Applying to humans, that's a bit more of an ethical minefield, but I think what will happen in the not so distant future is modifying stem cells that can be used. For instance, if you've got a blood-related problem, if you can get your bone marrow stem cells, which generate all your blood cells, and essentially get like a bone marrow transplant with these corrected cells, I think that sort of thing is going to happen, and that corrects the gene and should, in these sorts of cases, cure the disease pretty much after a bit of undergoing the transplant. And so that sort of thing will change cells in the adult, but because it's not changing what you're passing on to the next generation, it's limited to that person. And so like we do in mice, what we do in the mice is to change the embryos, and we change what can be inherited by the next generation. Obviously, we need to do that to be able to do our experiments. Whether that's necessary or desirable is going to be something which is debated. We already know how to eliminate a faulty gene that we know is in a family. That can be done by in vitro fertilization. So my suspicion is that will be the way to go. It's much less dangerous. It's already being done. It's not entering an area we don't know about. But of course, people are trying to modify human embryos, and I think that'll something that we have to grapple with in the future. But for the moment, it's proving to be a fantastic way of really putting the pedal to the metal on assessing what's happening with some of these mutations that we're pulling out of our genome screening approaches. Anyway, I will leave it there, and if anyone's got any questions, I'd be glad to take them. Thank you.
Thanks, Rob.